So today I will talk about work that we have been doing in my lab, but also use it to introduce different concepts in AI and in AI-assisted image analysis. And since I heard that there are different levels of expertise here, and it's very multidisciplinary, I tried to adapt and have more of a chronological perspective of what we have developed over the years in the lab, directly focusing on biological application and how we are developing the next tools right now. So the work I will be showing today is mostly the work from two extremely talented PhD students, Anthony and Frédéric, who have developed over the past years multiple approaches to make AI approaches better, more generalizable, and also adapted to our biological questions. My research lab is in Quebec City at the CERVO Brain Research Center, which is associated to Université Laval. And for students, but also research professionals that are here and who will be interested in participating to a summer school, we organize every year the Frontiers in Neurophotonics Summer School in Quebec in June, where you can test and try multiple microscope, many custom setup for a different neuroscience application. So today we will talk about AI and about quantitative and high throughput bioimaging. And why do we develop so much approaches using AI right now in microscopy? Well, one of the reasons is that AI models can be very versatile, and we have multiple tasks when we look at our images and we want to quantify what is happening. It can be very simple, such as detecting cell bodies, and we have many tools right now that are very good at doing so. It can be also classifying, segmenting, and also trying to do multiple of those tasks at the same time. We can have also tasks such as registration or image generation, increasing the resolution of the images. And then we want to develop models that are adapted to multiple modalities. For example, it could go from full brain imaging to the neuronal circuit in a zebrafish brain, to counting cells in mouse brain slices, looking at neuronal connectivity in cell cultures acquired on a confocal microscope, down to looking at subcellular structure with super-resolution microscopy. And this requires mostly very highly qualified experts to generate the data sets, to acquire the image, but also to analyze them. And ideally, we would want to have models that are able to perform as many tasks as possible on those data sets and be as generalizable as possible. So there has been multiple fields of development in microscopy, and I will name a few only. For example, single cell segmentation. This is something that we can do very well now, for example, with CellPose and SAM. We can also do beautiful things with enhancement, adding fluorescence channel in our images, enhancing contrast, restoring some images. There have been multitask pipelines that have been built by different groups where we can perform multiple tasks using different deep learning models at the same time. And then data-driven microscopy pipeline, adapting in real time to the sample and to the structures of interest. And many groups have worked on developing great reproducible pipelines that are contributing to making AI for microscopy more accessible and reproducible. In super-resolution microscopy, there was also a lot of work that has been done. We can think about single molecule localization microscopy, where we now have different algorithms that help reconstructing the images. Also image restoration, generation, and even super resolution, trying to generate high resolution images from lower resolution images or from very noisy images. And finally, different approaches of reconstruction that have given beautiful papers, especially in neuroscience. So this is the field where I'm working, and I call it often AI-assisted nanoscopy. And we have been developing different type of approaches. One of them is trying to improve the microscope while acquiring images. So optimizing microscopy parameters when we are taking the images, either to take better images or to help non-expert user to be able to use complex microscope, for example, super resolution microscopes. We have also integrated image generation into our acquisition pipeline in order to decide whether or not it's worth it to acquire a super resolved image. And if it is required, then make sure that we acquire the images in the most efficient way possible, reducing photon budget, reducing the photon exposition, and therefore augmenting our budget of good and super resolved images. Especially in living neurons, this is very important. And finally, we have developed also approaches where we can train a reinforcement learning agent to actually operate the microscope and to learn what is a super resolution microscopy acquisition and how can we adjust the parameters in function of the sample in order to acquire the best images as possible. But today I will not talk about image acquisition guidance. I decided to focus more on the work we have been doing for quantitative analysis. And I will show it in a chronological way of development we have done in the lab that also fits with some development we have seen in the field of AI. And when we started working on the first project with weakly supervised deep learning, the first experiments we did before publishing the paper were just after the UNet had been published in 2015. And we tried to apply it to our sample and to our samples. And you can see it took us a few years until we were able to publish. And one reason for it, well, there was a biology that was long, but also because back then it was way more difficult to have deep learning models that would generalize well to different experimental conditions, different cells, level of noise. And so we worked quite a bit to make our model more robust and better at assessing our data. So today we will talk about different approaches in machine learning, and I will focus on supervised learning and unsupervised learning. And we will try to look at different ways of reducing the expert annotation burden, making the model learn as much as possible without expert interaction in order to reduce biases, but also to reduce the time the experts are needed to annotate images. So for the next few slides, we will talk about a structure called the F-actin and spectrin periodical lattice that we can see only using super resolution microscopy. And it's a lattice composed of F-actin rings and spectrin filaments. And the F-actin rings are spaced by about 180 nanometers. And we were interested in understanding how this lattice contributes to cell function and how it reacts to changes in neuronal activity. And what motivated our work was the discovery of this F-actin lattice in 2013 by the group of Zhao Fang. And they discovered it first in axon. And a few years later, it was reported then in dendrites by many different groups, but with findings that were varying a lot between the groups. Some reported a mixed structure of F-actin rings and fibers, while others would see very, very clear F-actin rings. And what we could see is that the proportion of F-actin rings in the dendrites was varying, among others, with age. But other groups would also see those tiny actin fibers in the neuron. And we were wondering why there was such a diversity in structures, but also when we were looking at our images, they looked very complex. We had those small fibers that we could identify in the dendrites, but we would see patches, diffuse pattern, axonal rings that were very clear, and also F-actin rings in dendrites. And those patterns were always coexisting. So we first tried to use methods, or to develop methods that will allow us to analyze those F-actin rings and fibers that were the most prevalent structures that were reported by others. And the first thing we tried was to use classical approaches, Fourier analysis, threshold-based approaches, different types of really classical analytical signal processing approaches to see if we could analyze our signal. And one issue that we had was the high variability between cells, weeks, neuronal cultures. So our approaches would work well for 1 week, and then the next week it would not. So we were not able to find a way to analyze those structures in a reliable way that was robust over time. And our images were too complex also to analyze them all manually. So that's where we thought about using machine learning. And instead of using known rules, the goal was now to learn the rule, to train models to be able to learn using labels from our data what was the filters that were required in order to detect F-actin rings and F-actin fibers reliably across our full data set. What we wanted to do was take images like this that were treated with different activity modulating solution and be able to segment all the F-actin rings that we would see in the dendrites, but also at the same time the F-actin fibers. Because what we were seeing is that when we were stimulating the neurons, we would see an increase in the quantity of F-actin fibers, those very tiny longitudinal fibers that we can see here, are prevalent when we treat the neurons with an activity stimulating solution. So we had to find a way to perform this task, and one possibility would be to do precise labeling on the images. This is a very tedious task. It takes ages. At that time, I was a postdoc, so I was doing it by myself, all the annotation, spending hours annotating the images. And one thing that we would realize is that it was very variable from one person to the other, so I could not have help. I could not find help for someone to help me annotate the image because there were too much discrepancies between our annotation. The next idea we had was to annotate a data set with bounding boxes. We knew that those bounding boxes were not perfect, but we were hoping that the deep learning model would be able to learn from those labels in a weakly supervised manner and to learn what are F-actin rings and fiber and generalize it to the whole data set. So I annotated multiple images. It still took me over 100 hours to create the training data set with bounding boxes for F-actin rings and F-actin fibers. And then we trained just a regular unit which was starting with input images that were annotated and then trained to be able to produce segmentation maps. And this is called semantic segmentation because we are segmenting but also classifying the pixel for their, if they belong to the actin fibers class or actin ring classes. And there can be also overlap between the structures. So we start from a data set that was annotated with bounding boxes, and we get prediction from the network about the presence of F-actin rings and fiber. What we see is that the F-actin rings that are segmented in green here are replaced gradually by F-actin fibers if we increase neuronal activity. Big advantage of using AI to do this. It would take me about 25 minutes to annotate one image. Now I could do 1,500 images in 25 minutes, which was very good for my postdoc supervisor, who had always new ideas about new conditions I could test in the lab. So now I had a tool to be able to analyze those images. And with this, we were able to measure on many cells, different weeks, different cell culture, how F-actin ring will remodel into F-actin fibers with increasing neuronal activity. So here on the x-axis, we see the percentage of the area that is covered by what was segmented as F-actin ring, and here the fibers. At low activity, so if we block NMDA receptors, we have almost only F-actin fibers. If we stimulate the neuron in a LTP, long-term potentiation-like stimulation, we see that we have a shift to actin fibers, which is more important with a stronger stimulation of high potassium, and finally a very strong stimulation of glutamate glycine, which we can also use for long-term depression, completely change the F-actin rings into F-actin fibers. So we can see that with increasing neuronal activity, we have also an increase in the prevalence of F-actin fiber and a reduction of the F-actin rings. With this high throughput analysis pipeline, we were also able to analyze different conditions. So for example, to look at the dependence on the extracellular calcium concentration. And what we could see is that if we stimulate the neuron with high potassium, so this is the simulation of high potassium, but we block the calcium entry with cadmium, we completely block the remodeling of rings into fibers. On the other hand, if we add more calcium in our extracellular concentration solution, then we see that we have about only fibers when we stimulate the neurons with high potassium. So this was an example on how we can apply, in that case, a supervised deep learning approach with known features, manual annotation, in order to do some quantification in biology. Also helping us to better understand that actually all those structures observed were most likely related to different stages in the neuronal cultures, but also different activity level. So now I will, and that allowed us to make this model of the remodeling of F-actin in dendrites, which is not observed in axons, by the way. The axons were quite stable in that study. But the next question we had was, okay, it still took about 150 hours to generate the training data set for this unit. Is there a better way, a more efficient way to reduce even more the annotation burden that is put on the expert? And the next project we worked on was trying to go from the bounding box labels that still take 1 to 2 minutes per small image to do, to a very fast annotation process where we would ask an expert only to say if they see rings or fibers in the image. So we wanted to work with binary labels, and we applied that to multiple data sets, but today I will concentrate only on the F-actin data set. And so we wanted to develop a model that would be able to work with binary classification labels, but still produce segmentation maps, because that was what we wanted to quantify. So we used a very simple deep neural network, and what we wanted to have is as an input the image and the binary label. We pass the image through the network, and as an output, we want to have the classification, but classification of each pixel so that we can produce a segmentation map. So again, semantic segmentation, but this time using only binary labels. To explain how we did it, I will use an easy case of a modified MNIST data set where we have handwritten digits and we can have multiple digits on the same image. And the goal here is to produce segmentation map of each digits, but without telling the network where the digits are, just telling the network that there is a 0, a 2, a 7, and a 6. So we created this modified MNIST data set with a variable number of digits on each images, and what the network would get is just the binary labels associated with the presence or not of a class in the image. This was what was used as input, and then we decided to use the GradCam, so the grad and class activated maps of the network, in order to be able to use this output and create our segmentation map. And so first we look at the GradCam at the end of the layer, layer 8, and what we see is that the GradCam encodes the position of each digits, which is very convenient because already we're close to a segmentation map if we threshold this position. So just by using the GradCam of the last layer, we're able to have a core segmentation map. But if we look at the GradCam from every layer of our network, we can see that at the beginning, we encode very fine information about the structures, while at the end, for the last GradCams, we have more positional embedding. So that's what we wanted to use and to leverage, that we can detect high-level features at the end for the GradCam of the last layers and low-level features at the beginning for the GradCam at the beginning of the network. So we concatenate the GradCam to create a feature space, and then we need to combine this feature space to get our segmentation map. We tested different ways of combining this feature space, and in our case, what was working the best was principal component analysis, but you could use any other type of combination. We tried with sum and average too. And with that, we're able to produce very fine segmentation maps of the digits, and so without having to train the network with the positional mask, but only with binary label of the presence or not of the digits. And this works also on our biological data set. So we compared, so this is the original data. This is the very fine or precise labeling done by an expert that will take multiple minutes per image. And then this is the output of MicroNet, which is the network that we develop. And we compare it with a unit that I presented before with Mask RCNN. Both of them were trained with bounding boxes, and also with Elastic that was trained with a coarser label, which is only a scribble. And what we can see is that MicroNet learned very precise representation of the structure of interest. Actually, in frequency space, it was the method that was able to capture with the highest precision the presence of the actin rings in comparison to the expert labels. And we could do exactly the same for the F-actin fibers, and what we could see is that MicroNet would really learn to detect single fibers that we could use for further analysis. Okay, so the two first approaches that I showed today were using some supervision. We needed to have some expert labels, and for the two last approaches I will talk about today, we will shift to synaptic proteins and to more unsupervised training approaches. So the first One is fully unsupervised training, and where we wanted to analyze synaptic remodeling at the nanoscale and analyze synapses as a full population. So when we get our images of synapses, they are very beautiful, but also very complex, because we have a full population, heterogeneous population of synaptic structure, and also with different level of association between the pre and the postsynaptic site. Capturing the heterogeneity of the population is very important for us, as synaptic plasticity will not affect all synapses in the same way. So we wanted to develop a method that would be able to capture this heterogeneity and with minimal expert annotation or intervention from an expert. So we used a method that was developed by the group of Lydia Danglot called IC-Soda, and we adapted it so that it works in Python, and we called it PySoda. And here we have an example of an image that is segmented with the wavelet segmentation. And what we want to develop now is using PySoda is an unsupervised approach that would be able to find to characterize the synaptic population based on handcrafted features such as the area, the eccentricity, the solidity, the intensity, but also level of association between the pre and the post synapse, distance between the pre and the post synapse, for example. And the challenge that we have when we work with synapses is that we cannot control the amount of synapses we will have from each type. And actually, we have very often some synaptic subtypes that are that have a lot of examples, and some others that are very rare. And so we always have to keep in mind that if we analyze, for example, 2 types of synapses, but that their representation in our data set looks like this, with one of the subtype being the most prevalent. If we apply, if we look at the combined population, it would look very, most likely like the first population that is the more prevalent. And so if we apply a stimulation that affects only our minority population, unfortunately, if we look at the full population, we won't be able to detect it. So we need other approaches that will be able to capture these changes in some minority population in a data-driven manner. And we work in a multidimensional space because our synapses have multiple features such as the area, eccentricity, perimeter, coupling probability, so the probability of association between the pre and the post synapse, coupling distance. And so for this type of work, unsupervised approaches, for example, unsupervised clustering approaches, can be very powerful in order to find subpopulation within our data set in a data-driven manner. That's what we did, and we had 3 different conditions: LTD, LTP, so long-time depression, reducing synaptic strength, long-time plasticity, increasing synaptic strength, and a low activity condition as a comparison. We first projected our high dimensional space into in a lower dimensional space using UMAP projection. And after that, we did some clustering in our UMAP space. We then use the space of our UMAP, and we projected the different features that we had identified, such as the area, the eccentricity, over our UMAP to see how within our space, how the characteristic would change, and created synaptic barcodes. So for each of the clusters identified in our UMAP space, we created a barcode, and then using a silhouette score, we could determine how many, and hierarchical clustering, we could determine how many of those clusters were best representing our data set. We did that for different pairs of synaptic proteins and condition, and could, for example, for this bassoon and PSD95 pair, identify 12 synaptic subtypes with distinct barcode that we extracted from our data. This was completely without human supervision. This allowed us to create maps of our synapses in our images, and it was, of course, high throughput analysis. We could pass all our images through this analysis very fast, and then look at the features to interpret our result, because that's important that our data-driven approach also makes sense in terms of biology. So the first thing we look at is how synaptic clusters or synaptic subtypes are influenced by the activity level, and how different synaptic subtype will change following a stimulation. And what we can see is on the right side, we have all the clusters that have a high coupling probability. That means that there is a strong association between the pre and the post synapse. And those are increased following long-term potentiation stimulus, a chemical long-term potentiation, which is what we would also expect from biologically driven hypotheses. On the other hand, the cluster with low coupling probability, low level of association between the pre and the post synapse, were reduced in their percentage after long-term potentiation, which is also what we expect. We applied this approach to different conditions and then looked at how different synaptic subtypes will associate depending on different conditions. And so we will focus for now on the bottom of this graph, and what we see are the synaptic subtypes that were increased the most following this LTP stimulation. And we look at those subtypes and to which subtypes they were associated in another data set of synaptic scaling, where we had treated our neurons 48 hours with TTX, which is known to increase synaptic strength. And what we see is that there is a strong association with subtypes increase in long-term potentiation and subtypes that are increased after the TTX treatment in synaptic scaling. On the other hand, on the top here, we see that subtypes that are increased following long-term depressions are also the subtypes that are decreased during synaptic scaling. So subtype of reduced coupling probability, reduced association are associated together with two different data sets. And this was obtained fully in a data-driven manner and also across different experimental conditions and data sets. So for the last minutes of my talk, I will talk now about self-supervised learning, which is what we are working a lot right now in the lab. Again, with the idea of reducing human intervention, but increasing generalizability. And so before we talk about handcrafted feature that we had to define in our unsupervised machine learning approach to analyze our synaptic population. But now the goal is to learn those features, to extract representation from our STED images that will allow us to learn automatically those features without having to determine that before. So we work with foundation models, and you may have heard about foundation models in the past few years. They are becoming more and more important in microscopy. They have been published by many different groups in order to develop models that will be trained on very, very large data set and then fine-tuned on small pretext tasks, but that are able to perform extremely well on a large variety of data sets and tasks. And so to learn representation, we can use self-supervised learning. And the basic process behind self-supervised learning is to have a very large data set that can be fully unlabeled. So it can be all the images that you have in your lab or at your institute. And you will train an encoder and a decoder on a pretext task that you can imagine, for example, solving a puzzle, masking some part of your images, a task that you can have directly out of your images without requiring any labels. The goal of this part of the self-supervised training is just to have a deep learning model learn what is, for example, a microscopy image. What is a STED image? Not necessarily what is a F-actin ring or what is a synapse, but what are the most important features in a STED image. And then we can freeze a part of our self-supervised model and just fine-tune the decoder on a much smaller data set of interest on a downstream task, thereby reducing a lot the need for annotated data, but also increasing strongly the performance. One big data set that is known for pre-training is ImageNet. That's a huge data set that we use a lot in machine learning of photos from the internet and that are all labeled. And this could be a very good data set for self-supervised learning or to train foundation models. So we would use this huge data set, train a deep learning model to learn the features and the representation, and then we could fine-tune it on different pretest tasks. For example, segmentation of persons in photos, or segmentation in medical images, or some task in microscopy. And what we see is that the similarity between the image from ImageNet and from microscopy is quite low. And one issue that was reported on training foundation models with images that are completely outside the representation is that they don't perform super well, for example, on our microscopy images. So we created a very large training data set for STED microscopy. This data set is online. Feel free to download it and use it. There are more than 1 million crops, small crops of STED images from 40,000 STED images of a large variety of proteins, mostly in neurons. And we use this data set to pre-train our self-supervised model. We work on different self-supervised approach, but one of them is a mask autoencoder. And how the mask autoencoder works is just that you give this image to the network, but you mask a few regions of the image. And the task of the network is to learn enough about the features of those images to complete the masked areas. And this is a very common task for self-supervised learning. And so we can combine that with our huge STED data set and train a model to learn what is a STED image. And then I won't have the time to go into the details of all the tasks we tried on, but we could use it for multiple pretest tasks. For example, redo some clustering experiments, or work on image level retrieval or patch retrieval, so finding back images of tubulin in our data set, or finding certain types of synapses in our data set. We could work also on classification and on segmentation. And it was performing better than the other models we had trained before, because our model now has learned from so many STED images that it generalized much better to many different images. And we can use this also for image generation, and that's what I will present in the last 5 minutes. So for image generation, our goal is to represent the normal distribution of neuronal protein and use our self-supervised model to understand well what is a normal neuronal protein in order to be able later on to detect some anomalies, some outsiders. And so what we train is a generative model that will be conditioned on a self-supervised pre-trained encoder. So here there is a snowflake because it's frozen. So this is our model that we have trained on the very large data set. And now we fine-tune a model to be able to generate reliable STED images. And if we use the same self-supervised model, but that was pre-trained on ImageNet, what we see is that the reconstruction, so giving this original image, we ask the model to reconstruct an image. We see that it looks like a STED image, but the model was not able to capture the semantic of the structure that were given to it. However, if we use the same self-supervised training model that was pre-trained on STED images, now the model was much better in capturing the semantic, and we have generated images that are much closer to what we are interested in. And we can use that, for example, to do latent attribute manipulation. So what we do is that we want to manipulate the generated images to make them look more like F-actin, or, for example, more like tubulin. And it's like if we were taking a walk in our latent space, moving from tubulin to F-actin. And by moving in the latent space, so the space of representation of our network, we're able to generate images that look more and more like tubulin. So we start with F-actin, and then we ask we manipulate the latent space, and we ask the network to generate images that will look more and more like tubulin. And at the end, we have images that look a lot like our STED image of microtubules, and at the beginning, we were close to F-actin. This is only a toy experiment. This is not useful. But this was a proof of concept that it was working. But now we can use it for useful tasks. For example, if we want to understand what is happening in neurodevelopment when our synapses change at different time points in the culture. And we can manipulate our latent space and look at how different features of our synapses will evolve over time. For example, increasing their area and decreasing their density. So we could look using this model at the development of synapses across different time points. And finally, we can use such concepts and work together with computer scientists and astrophysicists in order to develop approaches that are very well known in the field of astrophysics, but apply them to the field of microscopy. And this is work with two colleagues, René, who is an astrophysicist, and Christian, who is a computer scientist. And what we are working on is how we can translate our approaches developed in microscopy and combine them with approaches developed in astrophysics to detect early signs of neurodegeneration. And so we go back to our synaptic population, and we want to go away from those handcrafted features because we don't want to induce any bias in our detection. And we think again about our heterogeneous synaptic population, where we could have the emergence of a new synaptic subtype, for example, during the process of neurodegenerative diseases. And finally, we want to develop a model that will be based on what I showed before, the self-supervised learning model, but that will encode normality. And what we encode here is what is the normal population and what is, for example, an aging or a model of neurodegeneration. And we try to find very subtle changes in the population. We are really at the beginning, but what I can show you is that it's working if we work with synthetic anomalies, but of course they are very different, so that was expected. But by using this approach, we're able to detect differences between normal synapses and synthetic anomalies. And now we can apply it to our data set of stimulation. And we can do exactly the same thing. So we extract the feature with our self-supervised model. We project that in a lower dimensional space. Then we apply our normalizing flow, which allow us to model what is normal and what is not normal. And now we see that here it was overlapping strongly, and we start to see a difference between the two populations. And if we look at at the population here on the top, now we can start seeing differences between synapses that were treated with the LTD stimulus without having to use handcrafted feature. And with that, I'm done, and I would like to thank Anthony and Fred, who did most of the work that I presented today.
Thank you very much for the nice talk. I was wondering if you ever tried a graph neural network on your synaptic data, because you have the distance information, and I don't know, maybe it would be nice.
Yes, Fred is working on that right now. And yes, it's a big challenge to be able to connect larger scale organization to very small scale organization. And we're trying different approaches, including graph neural network, to be able to capture this better.